Meiosis I is a process that divides a cell to produce a gamete. It contains 4 phases, each with their own factors that lead to the end division: early prophase 1, late prophase 1, metaphase 1, anaphase 1, and telophase 1.
Meiosis I begins with early prophase 1. During this process, the nucleus starts to break down and the chromosomes condense, making the sister chromatids visible.
Following early prophase 1 is late prophase 1. In late prophase 1, spindle fibers form and attach to chromosomes.
Synapsis leads to the exchange of DNA between homologous chromosomes, also known as genetic recombination. Before genetic recombination, the chromosome lines up in pairs of 2, also known as metaphase 1. The way the chromosomes line up is up to chance. The way one pair lines up does not affect another. This is known as independent assortment.
In the next process, called anaphase 1, the homologous pair is split by the spindle fibers. The process of splitting the pair is called disjunction.
In the final stage, known as telophase 1, the spindle fibers break down and the new nuclei form around the 2 different homologous pairs. The cell then splits in a process called cytokinesis, making 2 new cells. This concludes meiosis I.
Meiosis II is the process preceding meiosis I, in which the production of a gamete continues. During this phase, diploids are broken down into haploids. Meiosis II contains 4 different phases that lead to the end division: prophase 2, metaphase 2, anaphase 2, and telophase 2.
Meiosis II begins with prophase 2. In this process, the nucleus breaks down and new spindle fibers form around the chromosomes.
The next phase in meiosis II is metaphase 2. Synapsis occurs during this phase and the chromosomes are lined up single file at the equator of the cell.
Meiosis II continues with anaphase 2. In this phase, the centromeres divide, splitting the sister chromatids, making the diploid cells into haploid cells. The chromatids are then moved to the opposite poles of the cell.
The last phase is called telophase 2. In this phase, a nuclear envelope forms around each set of chromatids. The spindle fibers break down and cytokinesis splits the cell, ending division and creating a gamete. This concludes meiosis II.